Hello, my name is Dr. Thomas Hillen from the Department of Math and Start Sciences at the University of Alberta, and I do cancer research. And cancer is a very complex issue. It is as complex as a complicated jigsaw puzzle because we need so much information about the cancer. We need to know which organ is involved and also the stage of the cancer, stage 1, stage 2, 3, or 4. And then we would like to know the genetic profiling of the cells and also if they react to certain treatments and certain chemotherapies. And of course, we need to know how the immune system is involved. And on top of this, we need to know the status of the patient, how old they are, the sex of the patient, also if they have history, medical histories, etc. So it's a very complex system of lots of information which goes into cancer treatment. And mathematics is one of the languages that can be used to study complex systems. Now, cancer is no longer a death sentence, so treatments have improved quite a lot and many cancers can be treated very successfully. But there's one cancer, grade 4 brain cancer, also called glioblastoma, which is very, very difficult to heal, and mean survival times for patients affected with glioblastoma is less than 2 years. And this is where our research starts. So we use state-of-the-art experimental observations from a group in Heidelberg in Germany. This is a picture from a paper from Oswald, and that's a group of Frank Winkler. And this group won the Brain Award, or the Brain Prize last year in 2025. So what we see here is in green, a human cancer invading into a mouse brain. So these are experiments done on mice. And what you see in red are the blood vessels of the mouse. The characteristic feature here is that the glioma cells or glioblastoma cells are in green. They look like little beans. And they connect by these needle-shaped green extensions. And they are called tumor microtubes. And this is what our research revolves on. So the tumor microtubes, these little thin needles, are responsible for the growth into the healthy tissue, but they also allow communication between glioblastoma cells. And this observation has far-reaching consequences. For example, we could maybe think about finding treatments against the tumor microtubes. Maybe there are drugs which can destroy them, or maybe there are drugs which can prevent their organization or their communication. So there are many different ways this will help us to design maybe new treatments or successful treatment or treatment combinations for glioblastoma. Now here at the University of Alberta, I'm leading a team of talented young researchers, and we all work together on this important problem.
My name is Alexandra and I'm a third-year PhD student. I work on developing mathematical models for glioblastoma, which include tumor microtube-driven invasion and calcium signaling. The calcium signaling in glioblastoma networks is mediated by tumor microtubes. These mathematical models can then be used to further test treatments and develop treatments.
My name is Kyung-An Choi. I'm a postdoc working with Dr. Thomas Hillen and Dr. Hao Wang. I have experience in mathematical modeling of invading population, and I'll bring this expertise to this project to investigate the role of various treatment for glioblastoma. I plan to apply mathematical optimization methods to compare single treatments and combination therapies with the goal of identifying the best possible treatment options. These findings can then be used in mice experiments and ultimately to contribute to improving the treatment for human patients.
My name is Negar. I'm also a third-year PhD student. In my research, I became interested in oncolytic viruses, which is a promising approach to treat cancer using viruses. Currently, many viruses are being investigated, especially in combination therapies for glioblastoma.
We thank Brain Cancer Canada and all its sponsors for the generous support of this important research.